Hi everybody, this discussion is on citrate utilization. Bacteria need energy and nitrogen for their survival. In the normal situation, bacteria derive the energy from carbohydrates or sugars and they derive nitrogen from proteins or peptones. The citrate test is used to detect the ability of an organism to utilize citrate as the only carbon and energy source and ammonium salt as the only nitrogen source. The principle of the test is that some organisms have the enzyme citrate permease which allows citrate to enter inside the bacteria, and this citrate is broken down by the enzyme citrase to form oxaloacetate and acetate. This oxaloacetate is further broken down into pyruvate which serves as the energy source. Some citrate containing media are Cossers media. It is a liquid media with sodium citrate and ammonium dihydrogen phosphate as the main ingredients. They also have sodium chloride, magnesium sulfate and potassium dihydrogen phosphate buffer in distilled water. Here the citrate utilizing organisms will survive and the media will become turbid. Next is the Simmon citrate agar. The ingredients are the same as Cossers media, but agar is added to solidify the media, and bromothymol blue is used as a pH indicator. The media should be poured as slopes. No need to have a butt in the media. The original media is green in color, and on citrate utilization the color turns blue. The citrate media has an anion sodium citrate which is the only source of carbon and energy, and ammonium dihydrogen phosphate which acts as the only source of nitrogen. The pH is acidic, that is 6.8, and bromothymol blue is the indicator. The media appears green in this pH. The citrate utilizing organisms break down citrates and produce alkaline carbonates and bicarbonates. So also they will act on ammonium salt and release ammonia, which again gets converted to ammonium hydroxide. These alkaline products should raise the pH above 7.6, only then bromothymol blue will turn blue in color. Sometimes there is growth of bacteria but no color change in the media. Even this is taken as a positive test because only citrate utilizing bacteria can grow in this media. The alkalis produced were not adequate enough to change the color of the media. It is often seen that when we incubate such media for another 24 hours, the media will turn blue in color. The positive control is Klebsiella pneumoniae ATCC 13883 and negative control is E. coli ATCC 25922. We have to take certain precautions while doing this citrate test. While inoculating the organisms for special reactions, usually we inoculate the bacteria in peptone water, and a loopful of this broth is used to inoculate on other media like triple sugar iron, agar, mannitol motility media, urease, nitrate, etc. But for doing citrate, we have to take the bacterial colony directly on a straight wire and inoculate it on the media. If peptone broth is used, it will act as a source of nitrogen and alter the reaction. Next precaution is the bacterial load taken for inoculating the citrate media should be minimal. If a heavy load is inoculated, some bacteria will die and the dead bacteria itself can act as a source of carbon and give a positive reaction. While doing special reactions, always do citrate first. If we do all the other tests and repeatedly flame the loop, some amount of carbon can get deposited in the loop and this can again alter the reaction. Do not inoculate a sugar media like triple sugar iron agar before doing the citrate test. Some amount of sugar can be retained in the loop and this can again alter the test. Examples of citrate utilizing organisms are Klebsiella pneumoniae, some species of Proteus, Citrobacter species, Enterobacter species, Serratia marcescens, Providentia species, Pseudomonas aeruginosa, Acintobacter species, etc. When we talk about Salmonella species, some books say that Salmonella typhi is citrate negative. We have to be very specific. Actually, Salmonella typhi utilizes citrate in Cossers media. but it does not give a positive reaction with Simmon citrate agar. In some selective media used to isolate pathogens from the gut, we add sodium citrate so that it inhibits the growth of E. coli, which is the predominant bacteria in fecal samples. Examples of such media are DCA, that is deoxycholate citrate agar, TCBS, thiosulfate citrate bile salt sucrose agar, etc. I hope you have clearly understood about the citrate utilization by bacteria. Thank you.